So let's talk about the R packages. So R is a widely used programming language that we discuss for the statistical and data analysis. So as on the screen, you can see a several line of codes that I have written for installing packages. So in this video, we will explore different methods for installing R packages using the comprehensive R archive network, installing from the local files, fetching packages from the GitHub, and utilizing the Bioconductor repositories. So, packages are fundamental concept of core reusability in our programming. A package is a collection of functions, sample data, and documentation that describes how to use them. So, how to explore a simple R package? So, go to the CRAN database that should open right now. So, when we open the comprehensive archive network, then there is a lot of tools and libraries that are available here. So let's search for R to R, R project. So we can search for biostring package. Let's search for biostring. So here you can see a lot of R coding codes that are available. So for specifically using the packages, we can click on the package tab as well. So here you can see we can open the archive that contain all the packages that are available here. Here you can see the codes and the indexes of these codes. So CRAN is the primary package repository for or hosting thousands of packages. To install the packages from CAN, usually just open your RStudio. Here you can see the simple code. We have to read the Excel file that we are going to use. So here you can see if I just click here, they are saying that I already installed it. So they are saying that read Excel is available. In your case, it shouldn't show that. So just we have to just type in install dot packages and click on enter, and then just write the name of read excel. You can also just get these codes on my GitHub repository, and then just click control plus enter, and then they will be saying that one or more packages is about to. So I'm gonna click on no because I already have read excel that I'm currently working with. So read excel is already available. So in this way you can download and install or just install your CRAN package. So from installing a local file, you can download any. Sometimes you have to use the R packages that are stored locally. You have to download them and then perhaps from another source, or you just have that develop the code section by yourself. So you have to install that. So in my case, I'm going to use that command again, install.packages, and then locate where my package are available. So I'm going to use this command, and then I'm just going to install the string R that is already available for me. So string R is used for the genomic manipulation of data. So that is how you can download and install the package from your local file from using the GitHub repository. You know that a lot of tools that are available here are used by the a lot of other developers that are currently generating these. So using that GitHub link, you can also download and install your packages like Shiny. Shiny is a library that is used to create visual apps. So, for example, I created this app called Bioarray Explorer, which I can use to manipulate the metadata of metabolomics, genomics, and proteomics and transcriptomics. So here you can see is by my name, which is Rahman et al. So you can also use this for your analysis, which will be available soon online. And then you can use this Shiny to develop your apps as well. So first of all, you have to load the library of Shiny. For that, you have to just install Shiny on your system. I already have that, so there will be an error when you will be loading the Shiny file. So for that, you have to install the remotes package. When you have downloaded and installed the remote package, you can use the remote package to download to install the Shiny in your system, which is downloaded from your GitHub profile. So that's how you use the GitHub repository. So Bioconductor. Using the Bioconductor, you can download and install packages that is focused on your computational biology, bioinformatics, and your genomics field. So for that, you have to first download your BiocManager. BiocManager is the tool that is used to install your Bioconductor packages. For that, you have to install your BiocManager, and then using that BiocManager, you have to install some tools. For that, we are going to install the biostrings tool. So let's just wait while it runs the biostring. So biostring is used for the DNA strings to work with, like you want to reverse complement or complement the DNA sequence of an animal or plant. So I'm going to install the biostring by using the BiocManager, which I already exited the command. And then we're going to wait how much time does it takes to install the package. So that is a straightforward process, and understanding the different methods could be difficult for you. But for the thing is, you have to just install your packages to work with your files. For example, in our case, we are installing the biostring to work with the DNA string that we are going to reverse complement. So while we wait while it's in downloading and installing the package, so let's just wait if it's going to install. Right, I'm going to exit this command if I'm going to see biostring is already available. So loading package expect the following is package was built. So I already have the biostring package, so I'm going to just click on the DNA string and then reverse complement it. So here you can see it is reverse complemented. So the adenine is changed into thiamine or cytosine, and the guanine is changed into thymine. And that's how you get your 11-letter DNA string object, which is reverse complemented into your original DNA string. So that's all for your main bioinformatics and originally working with R and how to use the R packages. So what if you have to remove a package? So just like installing package, you just have to type remove dot packages. So that's how you can use that removal object to remove any package you want. And also there is a help tab that is available. So if you want to search for installing tool, you can also do that. And that's where you can get your help from the RStudio development company. And with this comprehensive guide, you can effortlessly install your R packages and unlock your new possibilities in your data analysis and statistical endeavors. So that's it for this video. In the next video, we are going to do the bioinformatics analysis of RNA visualization, and again move on to your WGCNA single cell and a lot more that is coming in our upcoming lectures.